Hi everyone, welcome to Unit 3, Cell Biology. Here is our overview of Unit 3. You will notice that there are only 8 lessons, so it's a pretty short unit. And we are going to start off with 3.1, Cell Theory and an Introduction to Cells. Remember for Unit 3, fill out your guided notes as we go through these lessons, but you also have a new vocabulary list, so make sure to use that list. And you will notice that for this first lesson, we only have 3 new vocabulary terms. And since we are not asking you terms from previous vocabulary warm-ups, that means that you only have 3 possible terms for your 3.1 warm-up. So we start off with Robert Hooke, and the old saying, seeing is believing, was certainly true for cells. Without instruments to make them visible, cells remained out of sight and out of mind for most of human history. All of this changed when a dramatic advance in technology, the invention of the microscope. In the late 1500s, eyeglass makers in Europe discovered that using several glass lenses in combination could magnify even the smallest objects to make them easy to see. Before long, they had built the first microscopes with these lenses, opening the path for cell biology. In 1665, Robert Hooke used an early compound microscope to look at a non-living thin slice of cork, which is a plant material. Under the microscope, cork appeared to be made of thousands of tiny empty chambers, and he called these chambers cells because they reminded him of a monastery's tiny rooms. Today we know that living cells are not empty chambers, but it took quite some time to discover that. In Holland around the same time, Anton van Leeuwenhoek used a single lens microscope to observe pond water and other things. To his amazement, the microscope revealed a fantastic world of tiny living organisms. Soon after, van Leeuwenhoek observations by scientists made it clear that the cells are the basic unit of life. In 1838, German botanist Matthias Schleiden concluded that all plants are made of cells. And the next year, German biologist Theodor Schwann, who also discovered a part of the neuron called Schwann cells, stated that all animals are made of cells. And in 1855, German physician Rudolf Virchow concluded that new cells can be produced only from the division of existing cells. And these are the discoveries that are summarized in cell theory. So cell theory states that all living things are made up of cells. Cells are the basic units of structure and function in living things, and new cells are produced from existing cells. So how do we find out about cells? Well, first, remember from that last slide, Robert Hooke was one of the first people to observe them under microscopes, but today's microscopes are obviously much more advanced than what Hooke was working with. So most microscopes use lenses to magnify the image of an object by focusing light or electrons. Light microscopes typically allow light to pass through a specimen, and they use 2 lenses to form an image. The first lens is called the objective lens, and it's located just above the specimen, and it enlarges the specimen. The second lens is called the ocular lens, and this magnifies the image further, or you can say that the ocular lens usually focuses. Some limitations of light microscopes are that light limits the detail or resolution of the images. Light waves are diffracted or scattered as they pass through matter. Therefore, light microscopes can produce clear images of objects only to a magnification of about 1,000 times. Another problem with light microscopy is that most living things are nearly transparent, and so we often use chemical stains or dyes to help view certain compounds or structures in the cell. In this picture to the right here, this is just a simple compound microscope. You've probably seen this one, but this is kind of the microscope that's most common in classrooms. So instead of using light, electron microscopes use beams of electrons that are focused by magnetic fields. Electron microscopes offer much higher resolution than light microscopes, and some are able to study cellular structures that are 1/100000th of a meter in size. Electrons are easily scattered by molecules in air, so the samples have to be placed in a vacuum to be studied with an electron microscope. So researchers must chemically preserve their samples, meaning that the samples have to be non-living tissues and cells for electron microscopes. So the 2 major types are transmission electron microscopes, or TEM, and scanning electron microscopes, or SEM. So here you can see a few different images. This one right here is the example of a light microscope, but that would be one that's stained. This one is a TEM, or transmission electron microscope, and this one is an SEM, or scanning electron microscope. So here is an example of a plant cell here, root tip, and this is within a transmission electron microscope, okay, and then here is kind of what the classic textbook-esque look of a plant cell is. So we use electron microscopes to see the structures inside a cell, then we create models to simplify. Darker images are more dense and lighter images are less dense. So cells come in all shapes and sizes. They can typically range from 5 to 50 micrometers in diameter. The smallest bacteria are only 0.2 micrometers across, and some giant amoeba can be 1,000 micrometers or 1 millimeter in diameter, which is large enough to be seen with the unaided eye. So down here, this is kind of that scale showing the different sizes. And then here you have that chaos, which is a pretty funny name, but that's that giant amoeba mentioned. All cells contain DNA, the molecule that carries biological information. And all cells are surrounded by a cell membrane, also sometimes called the plasma membrane. So those two terms are interchangeable. And cells fall into two broad categories, prokaryotes and eukaryotes. So you probably heard of these before, but here are two official definitions. A prokaryote is a cell that does not enclose DNA in a nucleus. A eukaryote is a cell that does enclose DNA in a nucleus. So here are some images. Okay, so here we have a prokaryotic cell, okay, and then you have two eukaryotic cells shown. So animal and plant cells are both eukaryotic. They all contain a cell membrane. So you see that here. They all have a cell membrane. And then for the animal and plant cell, you see the nucleus. Okay, so the nucleus is labeled. And then they also, for the eukaryotic cells, have these things called intracellular compartments, which we're going to get into more later on our eukaryotic cell lesson. But now more specifics on these two types of cells. So prokaryotes are generally smaller and we call them simpler when compared to eukaryotic cells. Prokaryotes do not have a nucleus or their DNA is free floating. They still grow, reproduce, and respond to their environment. Prokaryotes are unicellular, and bacteria are prokaryotes. Eukaryotes are generally larger and more complex than prokaryotic cells. Most contain dozens of internal structures and membranes known as organelles. The DNA is found in the nucleus. And eukaryotes, some are unicellular, okay, protists are unicellular and they're considered eukaryotes, and some are multicellular, so plants, animal, and fungi. So exit ticket, go ahead, pause here, and try out these on your own. So go ahead and check yourself. 3 parts of cell theory, and that is a vocabulary definition, so make sure to have that down. 2 things found in both prokaryotes and eukaryotes are DNA and a cell membrane or plasma membrane. Are bacteria prokaryotes or eukaryotes? And as stated, bacteria are prokaryotes. Eukaryotes contain DNA in a nucleus, and prokaryotes have free-floating DNA. Eukaryotes are generally larger and can be uni or multicellular, and prokaryotes are generally smaller and always unicellular. So essentially all multicellular organisms are eukaryotes. Are plant cells. Prokaryotes are eukaryotes, and plant cells and animal cells are both eukaryotes. So that's the end of lesson 3.1. Double check your notes and your vocabulary terms, and thank you for watching.